everyone, welcome to my channel Pure Biology, and today I come with another video that is the IP3 DAC pathway and PI3 kinase pathway. So there are three pathways, generally IP3 DAC pathway, PI3 kinase pathway and RAS-MAP kinase pathways that all are included or all are remained in the receptor tyrosine kinase pathway. Receptor tyrosine kinase. So this receptor tyrosine kinase, what is this receptor tyrosine kinase? And another pathway, just I have said that the RAS-MAP kinase pathway. So these two already I have explained in my previous videos, so you just check out that two obviously. And this IP3 DAC pathway and PI3 kinase pathway, today I will try to explain. Okay. And before going to this topic, I want to say thank you to all of you and who have already subscribed my channel. And this is my humble request to the mainly newcomers. Please you subscribe and share this channel if this channel is helpful to you. Okay, and just give your support to continue this channel. Okay, so let's continue. This IP3 DAC pathway is what? Many RTKs, that means the receptor tyrosine kinases, that can initiate the IP3 DAC pathway by activating isoform of phospholipase C. The phospholipase C, mainly gamma. Okay. So this pathway is actually dependent on the phospholipase C, mainly the phospholipase C gamma. Okay, because these DAC pathway, IP3 DAC pathway, these are activated only the presence of this. The SH2 domain. Okay, here this SH2 is what I have explained about this SH2 in my previous video, so you just check out. The SH2 domains of PLC gamma that bind to the specific phosphotyrosines of the activated receptor. And when the receptors become activated, when any ligand that has to be bound or that may be bound already in the receptors, then the receptors becomes activated. Okay. So what is happening here? The SH2 domain of PLC gamma that bind to the specific phosphotyrosines of what? The activated receptors. Thus the positioning enzyme close to its membrane bound substrate PIP2. Okay. And this membrane bound PLC gamma after activation cleaves PIP2 to generate two second messengers. One is DAC, another is IP3. And signaling via IP3 DAC pathway leads to an increase in cytosolic calcium and to activation of protein kinase C. So what is saying that? First of all, that here phospholipase C gamma is very important. Okay. Without phospholipase C gamma, this process is not possible because the mainly SH2 domain of phospholipase C gamma is very important, because this SH2 domain only bind to the specific phosphotyrosines. Specific phosphotyrosines of what? Of the activated receptors. So this binds to the phosphotyrosines of activated receptors. Okay. So here the positioning, okay, first I have to check one thing. What is saying that here when the binding is completed, so enzyme is closed. Okay, or position of the enzyme that is closed for what? The substrate PIP2. Okay. So here the membrane bound PLC gamma just after activation cleaves the PIP2. Achha, okay. So here PIP2 it has important role. Just after activation, this PIP2 that cleaves into two messenger. One is DAG messenger, another is the IP3 messenger. So here the signaling process is occurring through this, and that leads to increase in the calcium level in the cytosol and activation of the protein kinase C. Clear? That means if P activation is here completed, then PI2 that dissociate into two parts. What? One is IP3, another is the DAG. Clear? And if this DAG and IP3 formation is completed, then what is happening? Signaling process occurring, and this signaling process leads to the increasing level. This leads to increase the level of calcium in the cytosol, and obviously the activation of the protein kinase C that is also occurring due to the formation of this IP3 and the DAG. Clear? So here PIP2 just break into two messenger, and after formation of the two messenger, two things is occurring. One is the calcium level increase, another is the activation of the protein kinase C is occurring. Clear? Third is the PI3 kinase pathway. Here PI3 kinase actually catalyzes the formation of PI3-4 bisphosphate. Okay. So what is happening here? PI3 kinase that catalyzes PI3-4 bisphosphate or PI3-4-5 triphosphate. Clear? So after catalyzation of PI3 kinase, the two thing is possible, or two product may be formed. One is PI3-4 bisphosphate or PI3-4-5 triphosphate. Okay, and these are very important. Why? Because these are actual the sites for the signal production. Clear? Of what? The transducing proteins. So here various types of PI3 kinases are there, but some are activated by the beta gamma complex of trimeric G protein when GPCRS are activated by their signals. So it's possible only at that time when GPCR receptors are there or these are activated, GPCRS receptors. In that time, so here the PI3, these kinases are activated by what? By beta gamma complex of a trimeric G protein. Clear? Beta gamma complex of trimeric G protein, or very easily you can say by the G protein. Sorry, by the G protein. A primary binding target of PI3-4-5 triphosphate is protein kinase B. So main binding site or binding target is what? That is the PKB or the protein kinase B. Okay, a serine threonine kinase. So two things are possible, or two things are considered at the binding target. Target site you can say. One is the PKB, another is the serine threonine kinase. Clear? And besides its kinase domain, PKB contain, okay, here we have to learn about PKB, that PKB has kinase domain and also contains pH. What is pH domain? That is the P leg string homology domain. So two domains are there. One is that kinase domain, another is the pH domain. Okay, pH domain, and that tightly binds the 3-phosphate in PI3-4-5 triphosphate. So the two domains role is that to bind with 3-phosphate with triphosphate. In an inactive form, PKB is present in the cytosol. In case of rice PI345 triphosphate, that PKB binds to binds to them and localized in the cell surface. Again I repeat, generally PKB has two domains. One is the kinase domain and another is the pH domain, that is the P-dextrin homology domain. Okay, and what are the main role of the these domains? They tightly binds the 3-phosphate and the triphosphate. Triphosphate means PI345 triphosphate. Okay, generally when the level of PKB is very less, then they can remain in the cytosol. But when the rise of PI345 triphosphate is increased, then PKB binds with that and they come towards where? Cell surface. PI345 triphosphate generally recruit another kinase. What is that? This is the PDK1. This kinase name is phosphoinositide dependent protein kinase 1. And this binds to where? This is the plasma membrane. And binding to its pH domain, active PDK1 causes the phosphorylation. Okay, here just after binding, so first you can see that PI345 triphosphate and PKB both are bound with each other, and here what recruits again? Then another kinase, that is PDK1, that comes towards them. Okay, and after binding or after activating PDK1, the phosphorylation process starting. Okay, what phosphorylation? PKB's phosphorylations that get started. Okay, and when PKB fully activated, then dissociates from the PM for plasma membrane and phosphorylates many target proteins. Clear? So when phosphorylation is occurring, or you can say that fully activated, the PKB that dissociates from that is plasma membrane and phosphorylates to form many target proteins. So what is the role of actually this PI345 triphosphate? This 3,4,5 triphosphate generally what is seen here, that inactive form of PKB, when the PKB is not in active form, they remain in the cytosol, but when the level of this PI345 triphosphate increases, then PKB binds with that, and after binding PKB and triphosphate, then another protein, that is PDK1, binds to them. Okay, just after binding the PDK1, this activated, and this activated PDK1 that causes the phosphorylation of PKB. Okay, if PDK1 is not there, that means the phosphorylation of the PKB is not possible. Clear? So if PDK1 is not there, phosphorylation is not possible. Okay. So three are bound with each other, then PKB gets activated and the phosphorylation of the PKB that gets started, and so that phosphorylates many target proteins. Okay, so sorry, here one thing I have told wrong, that is the PKB actually activated and it helps in the phosphorylation of target proteins. Clear? So just it's activated and phosphorylates many target proteins. Clear? Okay. So that means here one thing is very important, that just binding and then phosphorylation. So binding and phosphorylation is occurring, that means the inactive form that change to the active form. Okay, so this pathway, the two pathways are very important of the receptor tyrosine kinase.